The overall goal of this procedure is to accurately determine the relative levels of post-translational modifications within preparations of total cellular nucleosomes obtained from mammalian cells. This is accomplished by first preparing high-quality homogeneous cultures of mammalian cells. Then, nuclei are isolated from the cells through mechanical disruption with a Downs homogenizer and centrifugation. The next step is to obtain intact nucleosomes by digesting the chromatin present within the nuclei and performing a series of hypotonic extractions. Lastly, the nucleosomes are interrogated using an ELISA assay with the appropriate controls to identify specific post-translational modifications. Ultimately, results can be obtained that show the relative levels of specific modifications present within nucleosome samples.
The main advantage of this technique over existing methods like Western blotting and chromatin immunoprecipitation is that a global picture of a number of nucleosome modification states can be readily determined. The method is much more sensitive and accurate than Western blotting and can be performed in most labs equipped for general molecular biology experimentation.
This method can help answer key questions in the stem cell and epigenetics field, such as what happens when major differentiation and reprogramming events occur.
To begin the procedure, first trypsinize the cells with 3 ml of trypsin per 15 cm plate. To freshly trypsinized cells, add 20 ml of ice-cold PBS butyrate, transfer the cells to a 50 ml conical tube, and centrifuge the tube to collect the cells at 1000 rpm for 5 minutes. Aspirate the supernatant and add 10 ml of ice-cold PBS butyrate to wash. Centrifuge the cell suspension again at 1000 rpm for 5 minutes. Remove the supernatant and resuspend the cell pellet.